All right, welcome everybody. Whoa, we're on air now. Okay, welcome everybody to this month's edition of Micro Seminar. Um, we are very honored to have Dave Baltrus from the University of Arizona giving a talk on the microbes inside of fungi. He will get into that, so I won't explain any further. Dave was did his undergrad at the University of Delaware, which is where Jen Biddle is now, but it was before Biddle. It was BB, and so they didn't cross paths there, but that's sort of an interesting irony. He did his PhD at the University of Oregon. Eric Allen, if you're out there, I know you're like screaming "Go Ducks" like right as we speak. There's the O. I did my postdoc at Corvallis, so this, whatever. Never mind. All right. The he did his David's postdoc was at University of North Carolina, which also overlapped with Jen Biddle. So there's a lot of cross-pollination there that maybe didn't happen, but now is. So without any further ado, Dave, I'll let you take it away, and we're all excited to have you. Thanks.
All right, thanks much. It's good to come to you guys from frigid Tucson, Arizona. It's supposed to be 102 today, which is cooler than it's been the last week or so, which is nice. So I guess I'll just jump right into it, and hopefully this will work. We good?
Stand by.
Yeah, you there?
Yep, works. It looks great.
All right, cool. All right, so what I'm going to be talking about today is a project that we started about, well, Betsy started it about 6 or 7 years ago, and I jumped in about 2 years ago, looking at bacteria living inside of fungal cells, so microbiomes inside of microbes. All right, so I want to start it off just kind of to get everybody on the same page and get you thinking like I think. I'll give you a little bit of history here. So 1976, which is, good Lord, like 40 years ago, there was a convention of the American Legion in Philadelphia. And at this convention, there was a mysterious outbreak of a flu-like illness. At the time, they thought this might be a flu, but it turns out that it was actually a new bacterial disease that was transmitted through the air conditioning system. It was about 25, 26 deaths, about 100 or so that actually got sick from it. The cause of this illness ends up being a bacterium called Legionella pneumophila, which one of the type strains is Philadelphia because it happened in Philadelphia. But Legionella's lifestyle is kind of interesting, and it plays into exactly why this outbreak happened. So Legionella pneumophila normally, oh, it's going through. No. I'm not used to Acrobat here. All right, so Legionella pneumophila normally lives inside of cells in amoeba. And it's somewhat of a pathogen of amoeba. It can just be hanging out there normally. But in the American Legion Conference, the amoeba was in the air conditioning system. There was Legionella inside the amoeba, and that got sprayed out to everybody. And the Legionella was able to then colonize everybody's immune cells. This ends up being somewhat of an interesting coincidence, and a lot of pathogens seem to do this, where they infect things like amoeba, and then they're also able to be pathogenic within your immune system because of miraculously your immune system and some of your T cells, I think, I'm not an immunologist, but I think it's macrophage, yeah, we'll go macrophage. Your macrophage end up looking a lot like amoeba, and so all of the systems that have evolved for Legionella to infect amoeba end up being the same kinds of systems that can be used to overcome your immune system. This kind of interaction has led to a variety of different research topics, this grazing resistance to pathogenesis. This is just a paper explaining, how there could be coincident evolution to both living inside of amoeba and then being virulent in your own immune system. So that just gets into, how amoeba can be host for bacteria. So single-celled eukaryotes can be host for bacteria. We've also turned this into a bit of a model system within the lab. So if some of you have done pathogenesis assays within Dicty or within amoeba, we use those as good hosts for some bacterial pathogens. And so there's this idea, we can infect single-celled eukaryotes within the lab with bacteria. It's probably happening out there a lot in nature. We just don't, I don't know, when I actually started this project, I didn't have a tangible sense that it was really present out there. It was really happening a lot. But the more you start to read and the more you start to look into kind of the literature, you see that this is really possible out there in the wild, where a lot of these single-celled eukaryotes are likely being infected at some point or another by bacterial cells. And then this last paper, I put it on there today because Joan Strassman tweeted it out because she couldn't talk about the GRC talks, but she tweeted out this paper from Matthias Horn's lab, basically looking at intranuclear, was my George Bushism there, intranuclear bacteria that can live inside of amoebal nuclei or other single-celled eukaryotic nuclei. So that's all of, by the way, of a long-winded introduction to say that microbiomes affect all eukaryotes. There's a lot of very hot research, hot literature out there looking at how microbiomes affect things like human systems. You'll see thousands of papers each week, it seems, that come out. But there's every eukaryotic system out there that's looking at either pathogenesis or symbiosis with bacteria is starting to deal with microbiomes and their effects on this. But when you start to look at it, there are single-celled eukaryotes and especially the amoeba, but a lot of the time also fungi that seem to have bacterial symbionts that persist for a while. There's a couple of different very well-known fungal symbiont systems where there's bacteria actually living in partnership or within fungi. The best of these might be the lichens. So the lichens are their partnership between either fungi and bacteria or fungi and algae. A lot of these lichens that are with the bacterial partnerships, the bacteria are kind of living outside of the hyphae, but it is a partnership where the symbiosis, both sides of the partnership get something out of this. If you actually look inside lichens, I saw a talk a couple weeks ago where if you look inside of lichens, there's also bacteria that can live kind of inside of the lichen as well, it looks like. And so you'll probably see some of that research coming out in a couple of years as well. So that's lichens. That's probably the most well-known of the systems. But those are usually thought to be extracellular fungal symbionts. There's a couple of pretty well-known model systems for intracellular fungal symbionts. On the left here is a fungus called Rhizopus. This one's a pathogen of rice, so this fungus is a pathogen of rice. But the pathogenicity within Rhizopus itself is determined by the presence of an intracellular bacteria within the Burkholderia family called Burkholderia rhizoxinea. This Burkholderia actually makes a toxin that allows the fungus to be pathogenic on rice. And there's a whole bunch of papers talking about the partnership between those where this is actually mediated by type 3 secretion, a little bit by type 2 secretion, and the bacteria actually transmitted through the spores. On the other side of it with the symbionts, so there's a lot of work within mycorrhiza looking at bacterial symbionts within the mycorrhizal fungi themselves. Some of these are Burkholderiae, so this Candidatus, Glomeribacter, Gigasporum. That's pretty much a Burkholderia that's present within mycorrhizae. And then there was also a couple papers last week on mycoplasmas that are present in a lot of these mycorrhizae that seem to mediate some of the beneficial effects of mycorrhizae on plants. But this is about what was, these are the main model systems, plus or minus a couple papers in a couple of other taxa that have been looked at in terms of which bacteria live inside of fungi up until this point. So I got into this project due to a partnership with actually my faculty mentor here at Arizona, Betsy Arnold, where she had started the work with her postdoc, Michelle Hoffman, a few years before I even arrived here. I'm looking at bacterial symbionts within a variety of Ascomycete fungi that they have acquired throughout the world. Just to give the overview of this talk today, I'll go in the background a little bit of those bacterial symbionts that Betsy and Michelle found in the endophytic fungi. So they work with fungi that are actually endophytes within leaves of plants. And so this is really kind of a Russian nesting doll kind of a thing where there's you have the plant, and then you have the endophytic fungi within the leaf, and then you have the bacteria within the endophytic fungi within the leaf. Kind of a nicely nested partnership there. That's always going to be echoed throughout this talk by these little Star Wars Russian nesting dolls. So the Wookiee, Chewie, is going to be the plant. Han Solo is going to be the fungi. Obi-Wan is going to be the bacteria, and then Luke is going to be the mystery partner that I mentioned at the end. So number one is going to be just an overview of these bacterial symbionts. Two will be the main question of the talk. Do these symbionts actually do anything, or are they just kind of passive partners that we see because we're looking for them? If you go out there and you look deeply enough at anything, you'll find interesting relationships. The question is whether these, whether what you find actually means anything, or whether they're just there. And then lastly, I'll talk a little bit about the genomic basis of the differences between the bacterial symbionts. So we've got a little bit of data just comparing genomes that might signal kind of what's going on here, although that's really speculative and kind of early at this point. But I'll mention it anyway. So this is, I made this in Keynote. It's in Acrobat, so we're going to lose some things here as it transmits over, but we'll just talk about number one, which is the symbionts of endophytic fungi. So this will be one of the only slides that actually deals with plants and actually shows plants within the whole talk. So all of the endophytic fungi that I'll be talking about today came from trees that are related to junipers. Most of them, that'll be the model systems I'll talk about later that we set up, actually came from this Platycladus tree. Not this actual one, this is a Wikipedia tree, but they came from this type of tree. And these trees were present either at the University of Arizona campus or in the Duke Forest outside of Duke when Betsy was a postdoc at Duke. And so these are, everything I'll talk about today is basically, you know, Betsy started it by going outside the lab, a couple feet in some cases, and actually just finding this outside the lab. So you don't really, you know, you don't have to go too far to find interesting stories, you know, anywhere really. Again, that'll be the only, this is the plant that a lot of these fungi are present within. So there's Chewy. Anytime you look at plants out in the wild, almost all plant tissue contains some kind of endophytes, and a lot of these are actually going to be fungal endophytes. Fungal endophytes are present within leaves, they're present within root tissue, and there's different species that will be present differentially in both of those types of tissue. And these fungal endophytes are thought to contribute to a lot of differences between plants. So the fungal endophytes are known to impact plant growth. They can act as plant growth promoting factors. They can help in plant defense and help for provide immunity against other pathogens. And a lot of Betsy's previous work to coming to U of A was actually dealing with the effects of these endophytes within on immune responses of plants. These fungal endophytes matter for plants, and pretty much all plants have them. The other thing that happens with plants, and I've watched enough Disney movies to know this is, you know, there's a whole circle of life going on here. So everything dies, and then it goes away and comes back again as a different life form. And so when plants die, their leaves are going to fall off, they're going to fall to the ground, and then those leaves have to be broken down somehow. And a lot of the time, it's actually the fungal endophytes can switch their physiology to actually start to break down and decompose some of these leaves. Leaves are mainly composed of cellulose and lignin, a bunch of other things too, but the cellulose and lignin are going to be some of the main components of what actually provides structural integrity to the leaf, and I'll talk a lot later about cellulose and lignin and why it's important that they be broken down. This is just a basic slide showing the life cycle of fungal endophytes. So a newly emerged leaf really isn't inhabited by anything, but it's going to be colonized by spores out of the air and sometimes bacteria out of the air. Over time, the density and diversity of endophytes within the leaf is going to increase to number 2. You actually get, there's some cool papers looking at spatial structure within the leaf and how you get diversity of different endophytic fungi throughout the whole leaf, many different types over many different parts of the leaf. And then, like I said, once the leaves die, they're going to fall to the ground, and they're going to be decomposed. And a lot of the times, we think of these fungal endophytes as being able to actually decompose the leaves. Then the fungal endophytes will sporulate, they'll get up into the air, and then they can infect newly emerged leaves. So there's this circle where the fungal endophytes actually undergo multiple types of physiology, where they're either growing within the leaf and providing defenses for the leaf, or once the leaves die, they can switch and start to break down the leaves. And we'll talk a little bit about maybe how these bacteria mediate some of these changes in physiology. So Betsy's work, and this is again coming before I even started at U of A. So Betsy Arnold and Michelle Hoffman published a paper looking at a variety of just doing 16S sequencing out of fungi that they got out of surface sterilized leaves. And Betsy and Michelle are able to show that across a variety of the fungal collection they have, and Betsy's collection is quite extensive. It's, you know, tens of thousands of isolates from around the world. In a lot of these fungi, they actually found what looked to be bacterial symbionts. They could get 16S sequences PCR'd out of the fungi, and these 16S sequences were not mitochondria. They were not anything that was, you know, weird that would be present there anyway. They actually look to be a diverse variety of bacterial sequences within the fungal hyphae. This is actually, if you want a lot of background, Hoffman and Arnold in AEM is going to be a good paper to go to for the background info here. I'll show you the tree of the sequences they got back in a second, but this is just a slide to show that they did fluorescent in situ hybridization to look at 16S specificity within these fungal hyphae. These are two different fungal types, and these little spots lighting up here are just going to be, those are, you know, a fish hybridization to bacteria within the hyphae. I get a question every time I've given this talk. I get the question so far, and I don't have the data to disprove it yet. But are these actually within the hyphae, or are they on the outside, and it's just a microscope plane thing? What I can say to that is that if you look closely, there's nothing kind of on the outside of the hyphae that you would imagine as if it were a microscope just changing the angle. And also, when we're isolating these hyphae, we grow them on plates, and if you just grow the fungi, you don't see any bacteria emerging until you kind of really stress them out. So if they were just hanging out on the outside of the hyphae, I would imagine that they would, the bacteria would grow kind of freely on the plates when we plate these out, but they don't. So hopefully in a few weeks we'll have the new fish to actually show when we reinfect that we can demonstrate this in Intrahyphal, but that data isn't arriving yet. So, and we've kind of struck out with trying some other fluorescent probes on it. So this, you know, long story short, there's bacteria living inside a fungal hyphae, and they've been able to sequence a variety of these out of it. So this is a tree from Betsy and Michelle's paper, just showing in the bolded, those are going to be the bacterial sequences that they found inside the fungi. These actually are all different fungal names, and then we color-coded these based on or coded them based on different bacterial phyla, or different types. So up here are going to be the beta proteobacter, gamma, alpha, and then gram-positives and a bunch of other stuff down here. Everything in bold is a bacterial sequence found within fungi. Everything not in bold are different taxa that they, Michelle and Betsy, got out of GenBank. That's going to be really annoying as it goes forward. Sorry about that. All right, so first point I want to make, in the yellow box up top, that's going to contain most of the previously seen bacterial symbionts. So that's pretty much the Burkholderia clade up here in yellow. Everything in purple are bacterial sequences that Betsy and Michelle found across these fungal endophytes. And so it's just kind of striking to me the diversity that they found versus kind of what was known previously. We've got Xanthomonas in there, or Xanthomonas-like bacteria in there. We've got Erwinia-like guys in there. We've got gram-positives in there, gram-negatives. It's all over the place. A lot of different diversity of sequences that they found within inside these fungi. And again, comparing that to the yellow box, this is kind of exploded the amount of sequence types that we were actually thought could be within the fungi themselves. Within a different project, within one of the grad students shared between Betsy and I, we found upwards of about 6 different bacterial sequences within a single fungal sample. And so maybe that's the limit, maybe not. But we think that, you know, there's definitely multiple types that we can find within single fungal samples, and maybe upwards of 6 or so. If you look at all of the endophytes around the world, again, Betsy's got quite an extensive collection. There's not bacterial symbionts within every single sample. It's kind of hit or miss. You know, some have 1, some have 2 sequences that we find. A lot of them, maybe, I'm just pulling a number out of nowhere, but I think it's around the ballpark. I think 40 or 50% of them, actually, we don't see any fungal bacterial sequences within them. So we're still getting a handle on the ecology of this and how prevalent these things are and how diverse they are. But I think there's a lot of potential there for saying that there's multiple bacteria living within inside these fungi, you know, upwards of 6 or so, we think. This is also not just all of these bacterial sequences are from leaf endophytes, leaf fungal endophytes, but we have matching sequences from root endophytes as well, and from seed-associated fungi. And so I don't think this is just a leaf endophyte thing as well. I think it's a lot of times when you look at environmental fungi, my guess is that you can probably find bacteria if you look closely enough with living what's inside those fungi. A couple of points to make. So it's going to be kind of annoying. Again, apologies for going to Acrobat, but it was the way that the screen share worked well. So a couple of points I want to make. You can see the names of the fungal taxa right here. So here's Phylosticta, here's Pistiloteopsis, I can never say that, but Pistiloteopsis, Alternaria. So these are the names of the fungal types. Within inside those fungal types, you see, these are very diverse classes of fungi. You see very similar bacteria, so there's almost no branching on this node right here. Very similar bacteria within inside diverse fungal types. And actually, now that we have whole genome sequences, I can tell you that at least in 2 cases, we've got bacterial strains from completely different fungi, so from a Pistiloteopsis and from a Microdiplodia, which is a different fungal class. We actually have nearly identical bacterial samples from that. I think there's, within those 2 bacterial strains found isolated from those diverse types of fungi, there's 1 major change between them that I'll talk about, and then 15 frame shift SNPs that may be real, maybe not, just due to sequencing errors. In the other case down here, again, we have similar fungi. These are Erwinia. I call them all Erwinia or Pantelia, but basically they're related to Erwinia or Pantelia from very different fungi. So Microdiplodia, Pistiloteopsis, these other ones as well. In this case, we've got 2 complete genome sequence samples where 1 strain is from Microdiplodia. I think the other strain is from a Pistiloteopsis. And they are, at the genome level, they have 5 SNPs, and 1 plasmid is different between them. So the take home from this slide, and what I'm trying to relate here, is that even within diverse fungi, we see very similar bacterial strains. So maybe it's just one kind of population of bacteria that can transfer between a variety of different types of fungi within the same forest. Although again, that's hand waving based on a couple of strains that we've isolated with whole genome sequences so far. And then the other thing to say, and this is just getting at that multiple types can be present within the same fungus or same fungal type. Even within Pistiloteopsis, so here's one isolate, here's another one. In this case, we've got Erwinia being present within it, and we've got a Xanthomonad kind of guy being present within it. And so even within a single fungal type, we can see very diverse bacteria within inside those hyphae. All right, so that's kind of the background and where I come into this. So my specialty, Betsy's specialty, is kind of fungal ecology. My specialty is bacterial genetics and bacterial genomics. So like I said, we've been able to isolate up to this point 11 strains and have the genomes completely sequenced. The remarkable thing about these genomes is that they're kind of unremarkable. They look like, environmental bacteria that they would be related to anyway. And one of the ways that I'll show this is basically using a graph right here where on the y-axis we have symbiont genome size, and on the x-axis we've got non-symbiont genome size. And so this, I'm just taking the 2 closest relatives from GenBank with whole genome sequences and kind of averaging out the genome size to the fungal symbiont. Typically, when you see very obligate symbioses, you'll see reduced genomes. So in John McCutcheon's work, you see crazy reductions in genomes within bacteria living inside of insects, living inside of the cells of insects. In some of the other fungal partnerships I mentioned before, I'll graph out a couple of these on here. You do see reduction in obligate symbioses, genome reductions versus the environmentally isolated guys. And so this is just a way to relate this. So I'm going to have the stars here are going to be previously characterized symbionts. One of these this is the Burkholderia rhizoxynica from one of the first slides, and then this is going to be the Candidatus glomeribacter genome. Again, both in the class Burkholderia. So I've color-coded these by the genus, so these are blue because they're Burkholderia. And what you can see, if you take these genomes compared to their closest relative, if there was no reduction, it would be along this 1-to-1 line, but you do see the genome size reduction within these 2 well-characterized symbionts. In the couple of PNAS papers from 2 weeks ago, the story with the Mycoplasma symbiont seems to be that there's not this reduction, but Mycoplasma are kind of weird and reduced anyway, so I'm not sure quite what to make of that yet. So we do have a couple of Burkholderia isolates, and just to show the difference between the isolates that we're dealing with and the obligate symbionts. Here again, it's a circle. That means it's one of our symbionts, and then it's in blue. It means it's Burkholderia. the genome that we sequence from the Burkholderia isolate found within the endophytic fungi isn't reduced at all. And this is a Burkholderia genome. It's like 8 megabases. It's huge. It looks just like the environmental Burkholderia genomes, even with multiple plasmids and multiple replicons. And so that genome's not reduced at all. And then when we look across the other bacterial taxa that we found, we don't really see any reduction either. So our endohyphal bacteria don't seem to have reduced genomes, which we think plays along with the idea that they're facultative symbionts versus obligative symbionts that have been seen before and have been characterized within fungi before. All right, so again, the acrobat problems here. So number 2 is going to be, now that we've seen these symbionts, do they actually do anything? If we clear them from the fungi, do we see phenotypic differences within the fungi of gnotobiotic fungi versus ones that have been naturally colonized? So we think that all of these bacteria are somewhat facultative symbionts because we can grow them all in isolation. The fungi can be grown in isolation, and all of the bacteria so far that we can sequence, we seem to be able to get types out of the fungi that can also grow in isolation from the fungi. And so what we can do, and this is just taking the platycladus isolate, so we surface sterilize this, get the fungi out of it. We can do live dead staining, 16S, PCR, fish, just to make sure there's bacterial symbionts in there. And then if you stress out these fungi a little bit, again, this is just a plate. You don't really see any bacteria growing there. There's really no yellowing outside of it. But if you stress out these fungi a little bit, say put them at 36 degrees, you do see endohyphal bacteria. That's what the EHB stands for there. You do see them popping out. So the first reason that we thought that these might be facultative symbionts is we saw this weird, or Betsy and Kayla and Michelle saw these weird fungal, what they thought were contaminants popping out in their long-term cultures. It turns out that these ended up matching up sequence-wise with a lot of the symbionts that they had seen. And so you can stress out the fungi, get the bacterial symbionts out, but if you just plate them out on just regular agar and put them at room temperature, you don't see crazy bacterial growth, again, which is why I think that it shows pretty well that these are intra-hyphal bacteria, not just kind of hanging out on the outside of the hyphae and growing wherever they would grow. So we can get the bacteria out. We can also cure the fungi of their symbionts just by plating them on a variety of different antibiotic cocktails. And then we can make sure that these are actually truly axenic by 16S PCR and fish, and making sure the signals before that they saw go away after we grow them on antibiotics. The other thing we've learned over time is that you don't really need these complicated antibiotic treatments. You can just grow these in rich media, and a lot of the time the fungi seem to kick out their bacterial symbionts on rich media. So that might be, I also get the question, why hasn't this been seen before? One of the ways that, back in the day we would isolate fungi, you plate it out on rich media. You plate it out and wait for it to grow and get isolates out of it. That plating out on rich media might actually have been selecting against maintenance of the bacterial symbiont. So that might contribute to why we're seeing this a lot more now versus isolates back in the day. Then the last important part of this, so we can get bacteria growing by itself, we can get fungi growing by itself, we can actually re-synthesize the partnership as well. So if we mix these two together, grind up the fungi a little bit, we can actually get the bacteria to retake into the fungi, and we can recolonize the fungi with bacterial strains. So now we've got the model system set up where we can actually make axenic fungi, test for physiological differences, and then re-inoculate with different bacterial types to see and really truly prove, this is my microbial genetics background here doing complementations. We can really truly prove if those physiological differences are due to the bacterial presence or just due to something else. So the two experimental systems that we've set up, just as a way to, everybody likes to have their model systems. These are going to be the ones that we set up for a couple different reasons. They start off with two different fungal classes, and so this Pistiloteopsis 9143 is going to be in the Cerdariomycetes. Microdiplodia 9145 is going to be in the Dothidiomycetes. And so these are really different fungi, both Ascomycetes, but in different classes of Ascomycetes. So that's kind of like humans to reptiles level of phylogenetic distance there. These numbers signify the fungal isolate, and so this is going to carry through to the bacteria that we talk about. So the endohyphal bacteria that we get out of these fungal isolates are going to have the same number. In this case, the systems we set up are, these are two strains of Luteobacter that we've isolated. Luteobacter 9143 we isolated from Pasteloteopsis 9143, and then Luteobacter 9145 we isolated from Microdiplodia 9145. Anytime the numbers match up, so anytime the bacterial symbionts come out of their original fungal host, I'm going to call that a native association. So up here 9143 and 9143 is native. Down here 9145, 9145 is native. Then we can re-inoculate, and so we're just going to re-inoculate, make Axenic Pasteloteopsis 9143, and then re-inoculate with either the native strain, so the native Luteobacter strain 9143, or the non-native 9145. And then we're going to see whether there's actually host range differences and physiological differences based on the specific bacterial symbiont of these fungi themselves. Coming back to the assays I mentioned before, so I mentioned that these endophytic fungi are known to change their physiology and help in breakdown of plant tissue, and so the assays I'm going to talk about are just going to focus on that stage of the life cycle. So we're going to look at the ability of the fungi themselves to break down plant tissue once it's dead. Betsy and Michelle also have a paper looking at some, in PLOS One, looking at interesting growth promoting differences between fungi with their symbionts and Axenic fungi, and it looks like there's a cool story with neither the fungi or the bacteria themselves can produce auxin, but if you put them together, the partnership produces auxin and promotes kind of plant growth promoting effects of it. So I think there's going to be cool stories of this partnership within living cells as well, but we're just going to focus on the dead cells, the dead plant tissue, and the degradative abilities here. So plant tissues are composed, a lot of them are mainly composed of cellulose and lignin, and so we're going to look at cellulase breakdown and ligninase ability. For the cellulase activity, it's pretty simple. It's a colorimetric assay. We just grow the fungi or the fungi plus their bacteria on maltose plates. Cellulose stains with a variety of different dyes. In this case, we're going to use Congo red. And then anywhere where the cellulose has been broken down, you'll actually see a clearing after you stain it. So in this case, all of this red out here is going to be the cellulose not broken down, and then this white part is going to be the clearing zone where the cellulose has been broken down. And the numbers I'll actually report are, you can see kind of how we measure it right here, or how Kayla measures it right here. We're just going to take the diameter of that breakdown area, and then we'll graph that out and normalize it for growth. So the first data point from these associations, so the naturally infected fungus with its normal symbionts, again normalized for 1, they're going to have a certain clearing of the cellulose up here. If we make the Axenic versions of that, cure them of the bacteria, we see the clearing zone go away. And so there's the first data point, truly showing that these bacterial symbionts do matter for fungal physiology. If you take them away, the fungi can't break down cellulose as well. I'll just show that in table form here. So with the naturally infected fungi in Pasteloteopsis 9143, naturally infected, it has cellulase capability, and the cured, it doesn't. So then we can look to go see, can we resynthesize it with its native symbiont? Can we resynthesize it with the non-native symbiont? Does this change the effects here? So this warms my heart as a, I like to do complementation assays with genetics. This is basically doing it with organisms here. But we can resynthesize with the native bacterial strain, and we can see this cured isolate regain its cellulase activity. And so, again, truly demonstrating that these bacterial symbionts and the Luteobacter are mediating some cellulase capability within these fungi themselves. And then it doesn't matter whether it's native or non-native, both the resynthesis strains, and we've done this actually, Kayla did this, 2 different resynthesized strains for each type. And so, and that's consistent, this data is consistent over each of those independent assays as well. And so this is not just a one-off thing either. And so it looks like, we can resynthesize and regain the cellulase activity. And that's just what it looks like in table form. So the cellulase activity is increased both by the native and the non-native symbionts. Just a little bit into we've just started doing the bacterial genetics of this, but it's kind of been fun because I've been back in the lab doing this. Just getting transposon Microbiomes of the bacterial cellulase activity. So this is what the Luteobacter looks like just growing on maltose plates. They're kind of goopy. I mean, they're xanthomonas, so they're going to be yellow and goopy basically. We can actually see cellulase activity underneath that colony. And we can get transposon mutants that actually knock this out. We just have a handful now. I would love to be able to tell you I know what the genes that are knocked out do. We know what the genes are. It's just that they're right now they're hypotheticals, or they're not really well annotated. And so they definitely don't look like cellulases. One of the mutants we have looks to be a transport system. The other is an outer membrane protein thing that doesn't have any cellulase activity, but it definitely knocks out. This is actually the outer membrane protein mutant. It knocks out the cellulase capability. And so I'm still kind of figuring out the genetics there, but there is we can get mutants, and it looks to be, we are going to find something novel here. I mean, if you look through the genomes of this guy, there's not any annotated cellulase activity or cellulases or anything that looks like they would mediate this breakdown. So hopefully we're going to find something cool with this, and I'll be able to report on that in a little bit later. So that's the cellulase activity. And now we're going to look at ligninase. And so ligninase is going to be the same kind of colorimetric assay. We're just going to stain underneath the fungal hyphae and then look for clearing zones of the lignin. This is actually indolin, but you know, it's the same chemical structure as lignin would be. In this case, we're going to have, I'll just show the data as I did before. So the naturally infected has a certain amount of ligninase capability, normalized for growth. And then it turns out when you cure the bacteria, the fungi of their natural bacteria, it doesn't actually change the ligninase activity at all. So that was kind of disappointing. There's the data, you know, just in table form, plus and plus. We can resynthesize with the native strain, and that doesn't change anything. So, you know, all right. But then the cool part is we can resynthesize with the non-native strain, and we haven't figured out what's going on here yet. But when we resynthesize with the non-native strain, the ligninase activity goes away. I was surprised by that. I'm still surprised by it. But again, we have independent replicates, independent resynthesis with the non-native strain. Something's happening here where the non-native strain decreases ligninase activity within the fungi. Yeah, it'll be cool to figure out what's actually going on there. We have no clue yet. So there, again, just in table form, ligninase activity is decreased with the non-native symbiont, but not with the native symbiont. So this shows their specificity to these symbiont effects, even within very closely related bacteria, the Luteobacter bacteria. So that's all in vitro assays, just looking at cellulose and lignin breakdown. What about rates of decomposition? Well, we can just go out into the U of A campus and get dead tissue from, you know, juniper trees or platyclatus trees. And Kayla did exactly that. We can bring it back into the lab. We can surface sterilize it. We can autoclave it. And then we can inoculate the different fungal mixtures into it. So either fungi with their native symbionts, fungi that are axenic, or fungi resynthesized with either native or non-native symbionts. We can follow this assay throughout time. And so basically, Kayla weighed these early on, right at the inoculation point. We parafilmed them, and then we waited, and I think she measured it over about 8 weeks or so. So, you know, she would weigh these over 8 weeks. We can actually measure rates of decomposition because there's mass loss. Where does the mass go? Well, it goes into water vapor, it goes into CO2, and that diffuses out of the plate, and you actually do see significant differences. You know, there is significant mass lost over time if you just leave these in the lab in Arizona on the benchtop. I'm going to show this percent mass loss on the y-axis here. Again, it's not much, but we've done the statistics, and any of the differences I've shown here are going to be statistically significantly different with non-parametric stats, and, you know, corrected for all the multiple comparisons. So the y-axis is mass loss. The x-axis is just going to be those different mixtures I showed before. So the naturally infected fungi, this is going to be 9143, the fungus we're talking about right now. The naturally infected one, it has a certain, you know, 2% mass loss. There's a certain rate of decomposition there. If we make it axenic, there's a statistically significant lower rate of mass loss. And so, you know, the in vitro assays for lignin and cellulase, that seems to follow through if we actually measure this total composite integrative measurement of decomposition just by measuring mass loss within a plate as well. So the bacterial symbionts matter for degradation. We can resynthesize with the native strain, and again, the decomposition ability could come back. And then if we resynthesize with the non-native strain, we don't see a difference from the cured strain. So there's interesting strain-specific, host-specific bacterially mediated effects of the symbiosis here on decomposition rates within the fungi. If you don't like just looking at graphs, here's kind of visual. I love this picture because it really demonstrates what's actually going on here. So here's just the control, again, just autoclave plant tissue, nothing inoculated into them over time. Now we switch to the 9145 fungus, but it's the same story. 9145 alone, this is 8 weeks later, it really doesn't grow, but 9145 plus its symbionts actually grows pretty freely over this dead tissue and actually decomposes the dead tissue pretty well. So again, I love this picture because it really demonstrates these bacterial symbionts are mediating growth, they're mediating decomposition. They truly do have effects here. What do the bacteria get out of the partnership? We are just starting to figure this out, and I am just getting the reagents in the lab this week to do the needed experiments. But I was, as a bacterial geneticist, I just wanted to figure out kind of what are the growth conditions of these bacteria, when can we grow them, when can't we? So the first thing I tried was just to grow Luteobacter, that strain that we've been working with, in minimal media and glucose, and it doesn't grow. It just kind of hangs out there, doesn't do much at all. But if we inoculate Luteobacter with the fungus in M9 minimal media with glucose, it grows well. It takes a while, but you do see bacterial growth. And so minimal media, I've talked before about how we can grow these bacteria outside the fungus. We've always been growing them in rich media, so either maltose plates with yeast extract or LB plates or rich media. And so it's not surprising that maybe they don't grow in minimal media because, you know, what is rich media, what is LB? A lot of it is just basically ground up yeast. And so whatever the Luteobacter might need to grow in minimal media, it's getting out of that yeast extract in the LB media. The same thing's actually happening here. We think the Luteobacter is getting either amino acid, maybe some peptides, maybe some other type of vitamin growth factor. It's getting from the fungus and allowing it to grow in that minimal media. So maybe there is some kind of limited auxotrophy here where the Luteobacter grows intrahyphally in order to actually be able to get some important growth effect. And lastly, just to kind of tease a little bit here, we have whole genomes for these Luteobacter strains and a variety of other strains, but we can use these whole genomes to start to piece apart maybe what the basis of those differences were that I reported a couple slides ago. So the two Luteobacter strains we've been working with, 9143 and 9145, they're really not different. They have roughly the same amount of genes. They don't actually have any SNPs different between them. Any single base pair change nucleotide SNPs. They have 15 or 16 frame shifts, again, which may be just part of the sequencing process and errors due to that. So some of this difference in gene calling and gene numbers is going to be due to, you know, those frame shifts changing the gene calling. The other difference we see here, so that takes about 20 or so of those genes that'll bump it up by that. There's still, you know, 50 or so genes to be accounted for, 60 or so genes to be accounted for there. So when we look at these genomes, we can align them all together. Again, no SNPs between them, but there is one region that differs in presence and absence between 9143 and 9145. So this region right here, this is just a malalignment. This region right here is present within 9143. It's completely absent within 9145 in the genome sequence. This ends up being a prophage. So it's about a 40 kb deletion in 9145 or a 40 kb insertion in 9143. It's a prophage with some 10 or so non-prophage genes that may be mediating these effects. So hopefully I'll be able to tease that apart in the future. The story's going to get a little bit more complicated, but I'm not going to tell you guys about that right now. All right, so if we just go back to that decomposition rate graph, here's a naturally infected fungus. Here's resynthesized with the prophage plus strain. Here's resynthesized with the prophage minus strain, and then the cured. So that's why this fourth guy comes into here. So it's you know, a fourth level of prophage nested within bacteria, nested within fungi, nested within plants here. All right, with that, I think that's about it. So just the conclusions, many endophytic fungi, Ascomycetes at least, that we've been talking about today, they contain a diverse array of endohyphal bacteria. These endohyphal bacteria all grow quite well in rich media without the fungus, and they don't have reduced genomes, so they don't look like they're going to be obligate symbionts that have been thought about before in terms of bacteria living inside of fungal hyphae. The model system we've set up is Luteobacter plus a couple of different fungal strains. So the Luteobacter strains themselves, seem to be able to modulate the ability of fungi to degrade plant material through either cellulase or ligninase activities. And then it seems like maybe the Luteobacter is oxytrophic for something that the fungus provide. My guess is it's going to be a B vitamin, although I should have that data in a couple weeks. So with that, I'm going to, this project would not have been possible at all without Betsy Arnold. Her and Michelle Hoffman started it, just looking for these bacteria living inside the fungi, and she's been, this is on my end, it's almost an unequal partnership where Betsy has done a tremendous amount here. So everything I say today, she's got her hands in it as well. All of the cellulose and ligninase work and the decomposition work was done by a master's student, Kayla Arendt, who was mentored by both Betsy and I. And then a variety of people in the labs have helped out tremendously with all of this. And then funding the complete genomes came from JGI, and then we have an NSF grant to look at the symbiosis. So with that, I'll take questions.
Nice, Dave. That was really interesting. You have quite a few questions, actually, from Irene Newton, who's been following this intently. I'm going to go back to the beginning. I'm going to do her questions in chronological order. And for those of you who are listening in, remember, tweet questions to hashtag #USeminar, or if you're logged in on the Google Hangouts, you can just pop up and ask a question. But the first question from Irene was, how specific are the associations? Well, actually, no, I don't even think that's her first. Sorry, first question. Fungi seem so promiscuous with regards to intracellular symbionts. Are all of these symbionts found within a single cell? So that tree you put up with all those different symbionts.
Yep. So we find them within single samples, not necessarily. We haven't found them yet, I think, within a single cell within a hyphae, but everything I've described where there's 6 different types within a single sample, that's within, we haven't done the resolution of, which hyphae they're found in. We're just kind of sequencing the sample. So I don't know that we have the data to say there are multiple types within a single cell yet, although I can see how that's possible, but we don't have the data to show that yet.
So you need to do like single cell genomics on single cells, inside single cells.
Basically, yeah.
Okay. Her follow-up question on that was, how specific are the associations between the fungi with these natural communities, the fungi and the bacteria? She says, is it like the heterotrophs found within cyanobacterial cultures?
Yeah. So again, a lot of the answers will be I don't have the data yet. I will say that I've tried reassociating some other strains I have in the lab, just kind of Pseudomonas strains that in them, and I don't get much. And so I'll get about 10 to the 5th or so logs of bacteria out of the fungi with the symbionts. With the Pseudomonas strains, I get maybe 100 or so out of them when I try to just reassociate them. And so I think there is, at least at that taxa level, there is a level of specificity. I haven't done the actual tests across all these isolates yet to know kind of quantitatively how much they differ, but I know that there's going to be, just at the broad view, there's going to be differences.
Okay. Continuing with Irene's questions, does the Luteobacteria symbiont have a preference for oxygen tension?
I don't know. That's a good question. It grows pretty well. Yeah, it grows pretty well just under oxygenated conditions. It doesn't like the biofilm at all too much. It does. It's a Xanthomonas, so it does throw out a bunch of like. I don't know if you guys have worked with Xanthomonas. It throws out basically xanthum gum, bunch of EPS. So it does that too. And the strain, if you let the strains go for a day or two extra in culture, you can actually turn the test tubes over and it's like really viscous. So I don't know if that mediates part of the oxygen changes as well, because that'll definitely change the viscosity of things. But aside from that, I have no clue.
Okay. Final question from Irene is, have you tried Mito-C treatment on the phage-containing bacterial strain, presumably to get it to try and pop out?
Yeah, so it's the more complicated thing I alluded to. So it's actually, it does pop out naturally. And it's I'm starting to think of it more in terms of Don Arnold's kind of work in Pseudomonas syringae, where I think it's present within the genome, and then it pops out maybe to a circular form that isn't captured in the whole genome sequencing. Because we can get strains that pop it out, and then we can get PCR products where it's not in the genome, but I can still PCR it from within the cells. And so I think it's actually popping in and out naturally, I think, at a pretty good clip. And yeah, I haven't tried mitomycin C though to get it to pop out anymore yet.
All right.
I mean, we haven't tried the plaque assays either to try to transfer it. But
she made a final comment that might stimulate some thought, which is that there might be some cool bacterial host range stuff to test with the phage, which seems to mediate some of the fungus interaction.
Yeah, that would be awesome. We have a couple of the other luteobacters. So we've got 4 isolates that we've got complete genome sequences of. The 2 that I talked about are really close, and then 2 outgroups that if you go back to the phylogeny slide, you can see the outgroups there. They don't have the phage, so only the 2 closely related ones have it. So it seems like we can do, we might be able to move it around a little bit and see what's happening there. But
was that a phylogenomic tree, or was that just a dendrogram based on like gene presence absence?
That was real phi, so it was whole genome SNP based phylogeny.
Oh, okay, okay. Yeah. So those things, and you said they have no real SNPs.
The 2 that were
91.
Yeah, those 2 have they have like 15 SNPs if you look at the genomes, but I'm not convinced that they're truly SNPs yet. These are the primers to test whether they're SNPs.
That's awesome.
Yeah.
Okay, are there any more questions for David here? What an interesting system. I think, let's see, anybody inside the Hangout want to ask anything? Okay. Well, that was fascinating, Dave. Thank you very much for sharing with us. We appreciate your contribution, and best of luck figuring out that prophage thing.
Yeah, hopefully we'll see what happens.
Yeah.
Thanks.
All right. Well, thanks, and we'll stop it there.